In this video tutorial, I am going to discuss about the primary structure of proteins. Now, primary structure of protein is simply the sequence of amino acid in a protein, right? So, the definition of the primary structure of protein is the sequence of amino acid. It is the sequence of amino acids in a protein. In a protein is called as primary structure of protein, right? Now, in biological science, why we need to study about this sequence of amino acid? The reason is that sometime abnormal amino acid sequence occurs, in the human body, and this can lead to many diseases. So, if we have knowledge about this sequence of amino acid, or in a sense, if we have a knowledge about the primary structure of protein, of normal protein as well as abnormal or mutated protein. So, if we know about the amino acid sequence of this normal protein and know about the amino acid sequence of this mutated protein, then we can better diagnose the disease. Right? And along with the better diagnosis, we can also better understand the disease process, so we can improve the treatment modalities, right? So, the primary sequence of amino acid, that is, suppose this is the amino acid, suppose these small circles are showing amino acid, then these amino acid are attached to its next amino acid by this linkage. This linkage is known as peptide link or peptide bond, right? So, all of these are peptide bonds and all these small circles are the amino acid. So, polypeptide chain, it is the unbranched chain, and how this peptide bond forms, that we are going to study. Okay. So, suppose this is one amino acid, this is its central carbon, this is the side chain R1, this is the hydrogen atom. On the right side we always write carboxyl group, and on the left side we always write amino group. So, this is first amino acid. Suppose this is second amino acid, again on the left side we have NH3 plus, on the right side we have carboxyl group, upper side I will write H and lower side R2. This is third amino acid, again amino group on the left side, carboxyl group on this right side, side chain R and hydrogen. Now, while making peptide bond, what happens? One oxygen and two hydrogens are lost. One oxygen from the carboxyl group and two hydrogen from the next amino acid's amino group. So, what will happen? H2O molecule will get released. Here also same thing will happen. One oxygen and two hydrogens are lost. So, here from here also H2O is lost. So, while making peptide bond, one water molecule is released. Okay. So, now what will be the structure? So, this R1 will remain unchanged, this H will remain unchanged, this amino group will also remain unchanged, but this carboxyl group, it is now in the form of C double bond O, that is, only single oxygen atom remains. About this amino group, what will happen? This is N and H, only single hydrogen atom will remain. The rest structure, this hydrogen will remain unchanged, this side chain R2 will remain unchanged, but from this carboxyl group, what will happen? One oxygen is lost. So, now here again there will be only one oxygen, and from this amino group only one hydrogen will be there. So, NH will be there and rest structure will be unaltered. So, C, this is R3, this is H and this is COO minus. So, here as an example I had only shown you 3 amino acid peptide chain, right. So, now what will happen? There will be a formation of peptide bond over here. So, this portion, it is peptide bond. This is also a peptide bond, right. So, these both are peptide bond. This peptide bond is the strong bond, it is the covalent bond, strong bond. So, if we want to break down this strong peptide bond, we have to use extra force. Okay. So, to hydrolyze this strong bond, we have to use strong acid or base. Not only strong acid or base will be sufficient, but we have to use it at a elevated temperature, and this condition should be provided for the prolonged time. So, this much harsh condition is required to break down this peptide bond. Okay. Why? Because it is very strong bond. Now, on this side we have free amino group. This is free amino group, and this is free carboxyl group. Free carboxyl group. Now, focus on this central amino acid. See, in this central amino acid, what is happening? From one side 2 hydrogens are lost, and from another side 1 oxygen is lost. So, each of the central amino acid molecule, it is 1 H2O molecule is lost. Okay. So, that means this is whenever this central amino acids occur in the polypeptide chain, this amino acid is not in its full form, but some trimming has been occurred. From one side 2 hydrogen and from another side 1 oxygen is lost. So, whenever such amino acid occurs in the peptide chain, we do not call them amino acid, but instead we call them as a amino acid residues. We call them as a amino acid residue. Why residue word is used? Because it is leftover part of the amino acid after water molecule is lost. Now, from this amino terminal amino acid and from this carboxy terminal amino acids also some part is lost. Okay. So, essentially all the amino acids are not in their complete form in the polypeptide chain. So, here we call all the amino acids which occur in the polypeptide chain them as a amino acid residues, right. So, now next we will discuss about the naming of peptides. So, next we will discuss about the naming of peptide. Naming of peptides. Now, as an example, I had taken simply the 3 amino acid peptide chain, but when there are many amino acids are there, in the peptide chain, we call them as a polypeptide, right? Polypeptide. Now, whenever we write about the polypeptide, we always write amino terminal or the amino group on left side and carboxyl group on right side. And remember, amino group, it is also called as N terminal, and carboxyl group, it is also called as C terminal. So, whenever we want to read or write about the polypeptide chain, we always write from them N terminal to C terminal. Okay. So, for example, suppose one polypeptide chain is there. The first amino acid on the N terminal side is the glycine, the second amino acid is the glutamate, and the third amino acid, it is the tryptophan. And we want to write this amino acid sequence. So, we have to always write N terminal first on the left side and C terminal on the right side. Now, while giving the name of this polypeptide, we have to follow certain convention. See, all the amino acids either end with INE or ATE or AN. So, whenever we want to name any polypeptide, what we have to do? We have to remove this suffix INE or ATE or AN, and this suffix should be replaced by YL. Okay. So, YL should be there, but this rule applied to all the amino acid except for this C terminal amino acid. So, let us write the name of this polypeptide. So, instead of glycine, we will write glycyl. So, instead of INE, I will write YL. Instead of glutamate, instead of 8, I will write YL. Okay. So, glycyl, then glutamyl, that becomes glutamyl, and this tryptophan, as it is C-terminal amino acid, we don't have to replace its suffix. Okay, so I will write over here tryptophan. So the name of this polypeptide is the glycyl glutamyl tryptophan. Okay, so this is the naming convention of this peptide chain. Now we will see the characteristics of the peptide bond. Characteristics of peptide bond. We know that the peptide bond is the bond between two amino acids. This is one amino acid 1 and amino acid 2. This bond is with the carboxyl group of one amino acid with the amino group with another amino acid, right, that we had seen just a moment before. So this bond is between this C double bond O and NH group. This is the peptide bond. Now what happens? Typical C single bond end, typically this single bond, if it is there between carbon and nitrogen, its distance is 1.49 angstrom. Whenever typically there is a double bond between this carbon and nitrogen, the distance between this carbon and nitrogen, it is the 1.27 angstrom. Now why? Because this double bond is stronger, so this both atoms will be near to each other. So here less distance, but whenever single bond is there, that time there will be a less force as compared to this double bond. So the distance is more. When in polypeptide chain, this distance, this peptide bond's actual distance was measured, this distance was 1.32 angstrom. Okay, 1.32 angstrom, which is little bit lesser than the single bond, but more than this double bond. That means this peptide bond, it is neither single or nor double bond. Okay, it is between that. So we call this peptide bond as a partial double bond. Partial double bond. Peptide bond is a partial double bond character, and whenever there is a double bond, that bond is always rigid and planar in the same plane. So what does it mean? It means it is the partial double bond. So there is no any rotation can takes place over this. Okay, why? Because it is a rigid bond. So let's try to understand with one figure. So again, this is one amino acid, this is C double bond O, on this side we have NH3 plus, here we have R1 and here we have hydrogen. This is second amino acid alpha carbon, here we have NH, here we have carboxyl group, here we have hydrogen, and here we have R2, and this one is the peptide bond, right. Now what happens? Around this bond there cannot be any rotation, okay, whereas this is single bond, so rotation can take place over here. This is also single bond, so rotation can take place over here. This is also single bond, so rotation can take place. This is also single bond, so rotation can take place. Now you might be wondering why I am writing R1 over here and R2 over here. The reason is that this side chain R group, these are the bulky group. So if both occurs on the same side, then there will be too much congestion of atoms over here, which is thermodynamically unstable. So that's why rotation takes place at this location or at this location, and so this side chain R1 and R2 occurs on the opposite direction of this peptide bond. And whenever there is such opposite direction around this partial double bond, we call it as a trans configuration. Trans configuration of this double bond. Okay, why we call it as a trans configuration? Because side chains are on the opposite side of this hypothetical double bond. Okay, so this is the characteristic of peptide bond. So I hope that everything is clear about the primary structure of protein, their naming of peptides and characteristic of peptide bond. If you have any query or confusion, please write it down in the comment section below. Thank you.